So, a very good evening, all of you. Uh, can you hear me? Can anybody just put a message if I am audible? Okay, that's good. So, a very good evening, all of you. Uh, can you hear me? Just put a message if I am audible. So, I think we can start now and hope I'm audible to everyone. If there is any issue, please, please let me know if there is any issue. So I think we can start now and hope I'm audible to everyone. If there is any issue, please, please let me know if there is any issue. I think we can start now and hope I'm audible to everyone. If there is any issue, please, please let me know if there is any issue. Okay, so this is regarding to the course or what we are going to do. Uh, just give me 2 minutes. I think voice is echoing. Just give me 2 minutes. Okay, so this is regarding to the course or what we are going to do. Uh, just give me 2 minutes. I think voice is echoing. Just give me 2 minutes. Okay, so this is regarding to the course or what we are going to do. Uh, just give me 2 minutes. I think voice is echoing. Just give me 2 minutes. Okay, so this is regarding to the course or what we are going to do. Uh, So, good evening, everyone. This is the presentation which we are going to discuss today. So, before beginning with the actual presentation, I would just like to take you through what I'm going to do here. Basically, I have been getting a lot of mails and inquiries regarding some general understanding or general knowledge related to formulation development. So, as informed in the earlier webinar, I will be arranging at least 2 days a week. I will be arranging webinar. So now in this webinar, what I am going to do, uh, just a minute. Sorry, just a minute. So basically, this whatever series or tutorials we are going through, in this tutorials, I am going to take you through how we develop a formulation development, and we will work overall on how we develop a formulation development. So I would just like to inform that this series or tutorials are meant only for freshers or someone who is just entering the industry or who is having 2 to 3 years of experience, and maybe he wants to upgrade, he or she wants to upgrade the knowledge. So this is meant only for them. Might be experienced people might be finding this too basic as of now, and might be it not fall to their expectation. But thing is that I have received a lot many of requests in my inbox where mostly students or freshers, they have contacted me and asked me about guidance and all, and then I have been getting n number of questions also. So I thought this is the best way I can get in touch with you all. So yeah. So basically, this will not be something very complicated or something. If you are an experienced person, more than 5 years, 10 years experience, might be you might find this information too basic for you. But anyone who is interested is always welcome to attend these sessions. What I'm going to do, I have made a presentation so for today's session, and then next session, maybe sessions, I'll take you through this presentation about general formulation development routine. Once that routine is developed, then in the future tutorials or classes, I will take you through literature search, how literature search we do. Then with practically how we develop a formulation. Then we will one by one discuss the guidelines which comes effective for a formulation development. The guidelines will be from majorly from US market, Europe market, and MHRA market. Those guidelines we will discuss, and over the period of time we can have some interactive sessions related with it. uh, so this is the main agenda. So maybe I think 2 to 3 months I'll continue where you people can just have a broad idea, and maybe I'll practically show you how to do literature search, how to search the FDA websites, what information needs to be taken from freedom of information, what information needs to be taken from Orange Book, how to search for the patent related to it, some practical trainings and all I will be giving moving forward. So to start with, this will be the presentation which we will be working. So I'll start with the presentation now, and I'm sure I'm audible. If there is any problem related with my voice or clarity, just put a message and I'll get back to you. Sound. Uh, okay. I think Asim Kumar De, he's having sound. Okay, let me see. Let me see what I can do. Just give me a minute. Actually, my headphone has malfunctioned. So I'll be trying to be a little loud. Maybe you can hear it. So please put a message if you are able to hear what I'm saying. I'm just going through the message once. Uh, okay. Okay. So now I'll take you to the presentation. Maybe this is a 25 pages presentation, but maybe we will not be able to complete the full presentation today. Uh, but we will definitely complete it in next 2 sessions. So can you view this presentation? Can someone just put a message that you are able to view this presentation? Is the presentation audible? Sorry, can you see this presentation? Okay, the presentation is visible. So I'll now start with the presentation part. Uh, so basically, this presentation is just to give you an idea about how we go for a formulation development. overall what is the life cycle of a formulation development. So basically it will be related to comprehensive R&D activities from the initial concept when the project is developed to its market stage. Basically, it consists of four important parts: pre-formulation studies, quality by design, regulatory compliance, and bioequivalence. So this is an overview of generic development. So as you know, whenever we develop any product where the innovator is already there in the market and we are mimicking it and we are matching it with bioequivalent studies and all, it is called as a generic drug development where innovator is already there, sufficient work of innovator has been done, and we are just matching the blood profile with innovator. So that is what is called as generic drug development. Bioequivalence to brand name drugs. Brand name is of course the innovator, and then safety, strength, and quality. We match the innovator in this aspect, and they follow a systematic R&D process wherein the key characteristics are the API. Then we make identical strength, dosage, and administration route, and bioequivalent performance. So this regulatory pathway is called as ANDA. So ANDA is nothing but abbreviated new drug application. Uh, Abbreviated new drug application. Why is it abbreviated? It is abbreviated because the development time has been abbreviated, shortened. So whenever any innovator develops any molecule, usually it takes 20 years, 25 years for development, where they start the development work and they do they synthesize the molecule, then they characterize the molecule, then they perform the toxicity study, then they give to human volunteers, then they do the phase 1, phase 2, phase 3 clinical studies. Once the drug is safe, they put it in market. So these overall cycles take 20 years, and the innovators come in market. And then when we want a similar product in the market, in that case, what we do, we don't have to do all this 20 years of hard work. We have to just make a product and we have to do a clinical bio study with the innovator. And if we comply to the bio study, we can manufacture our product as generic. So maybe for our development time, it might just take us 2 years at the max or 3 years at the max. So this development cycle is shortened, and that's why it is called as abbreviated new drug development. And this ANDA terminology is used only for the US market product. In other market, the name keeps on changing, but overall, it is just the generic development product. So when we start any new product in R&D, the R&D life cycle has different phases. Discovery and target identification. So discovery and all is not what we do. It is usually the innovator who is doing. Target identification is identifying a particular molecule already available in the market and whether we can make a generic or not. That is the first phase of any product. Once we do that, we do the pre-formulation studies and we do the API characterization. I will walk you through these details of this as we move forward. Then we do the formulation development by applying QBD techniques. In this, we develop a bioequivalent product where our product pharmacokinetics matches with the innovator product pharmacokinetics in the plasma of the volunteers, and the parameters which we check are Cmax, AUC, and AUC infinity. Then once our product is proven bioequivalent, we develop a process, we scale it up, and then we go for bioequivalence and regulatory submissions. So this is the overall rough development cycle. Now I'll move to the next slide. So what is the regulatory framework and FDA requirements for generic product. So whenever we file any product as generic, depending on the market which we are filing, we have to answer or we have to comply to different regulation. For example, just now we discussed the ANDA pathway. So ANDA is when the innovator is already there and you are trying to make a generic product in the US market, it is called as ANDA. Apart from it, if you are intending to market the product in Europe, you have to follow the guidelines of EMA, European Medical Association. If you want to market the product in Canada, it will be Health Canada. If it is wanted to market it in UK, it will be MHRA. If Australia, it will be TGA. And then ICH. ICH is International Conference of Harmonization. These are the guidelines which we have to follow, irrespective of which market we are putting the product. There is just message. Can you click on slide share? A slide can be screen. Okay, I'll do it. I'll do it. Sorry, I've forgotten to make it slide share. I'm putting it on slide share mode. Okay. Okay. Uh, so then the key regulations are 21 CFR 314 for ANDA, 21 CFR 320 for bioequivalence, 21 CFR 210 and 211 for CGMP. These guidelines you have to follow at any cost. Then our regulatory focus, whenever we develop a generic product, our focus is majorly on demonstrating safety, efficacy, and quality via bioequivalence. Quality in bioequivalence means we have to manage the innovator characteristics, pharmacokinetic parameters for our product. So the primary requirement for any ANDA for US market is bioequivalence study data. The BE data has to be there. You have to first match the BE. Then CMC. CMC is a typical terminology which we use for usually the ANDAs where C stands for chemistry, M stands for manufacturing, and C stands again for control. Chemistry, manufacturing, and control. Then labeling and packing, facility CGMP compliance. This is a standard requirement for any ANDA to have. So now when we talk about BE criteria, so BE criteria, the criteria, whether the study is passing or not, the criteria is that we should have a 90% of confidence interval between the values of 88 to 125 for AUC and Cmax. So this is something which is statistical. It is basically a comparison when we compare our Cmax with the innovator Cmax and then we put the statistics. So the value should fall in between 80 to 125 for both AUC and Cmax, and then only you can say your product is bioequivalent. Of course, there is a relaxation to this limit when the product goes or when the product has high variability. This we will be discussing when we go through some advanced presentation. Right now 80 to 125 is the limit. So AUC is nothing but the area on the curve, that is the total drug exposure. Cmax is the peak drug exposure. Then as I told you, special cases. So in case of NTI, for example, a drug like levothyroxine, the limit is very tighter, that is 90 to 111. I will explain to you why it is tighter when we go there. And for a highly variable drug, it is a scaled criteria where the limit can be relaxed to 70 to 135. We will discuss it when we go in advance. Then the quality standards. So whenever you are manufacturing any ANDA, you have to have a manufacturing and process validation. You have to have proper quality control systems, proper stability program at the plant, and then you have to have sameness attribute, that is the matching of Q1, Q2, Q3. This is Not a standard criteria. In certain criteria, you have to match. In certain criteria, you can be totally different. When we say Q1 and Q2, it is qualitative and quantitative and microstructural. So I will move to the next slide now. I think there is still one more message. Okay. 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 Just give me a minute. Slide is not moving. I think. Okay. So ICH guidelines and global harmonization. So whenever we develop any product in R&D, we have to follow the ICH guidelines and global harmonization. So when we follow ICH guideline, majorly we follow the ICH guideline usually for the stability, but apart from stability, ICH guidelines has guidelines mentioned for analytical validation, for the development, that is Q8 to Q12 guideline, and then also the guidelines for good clinical practices and statistical principles. All these guidelines are covered by ICH guideline. And then apart from this, apart from this, safety guidelines are there, which is usually taken care by clinical. Efficacy guidelines are also taken by clinical. And then the multidisciplinary guidelines are also there, which are also taken care by a common technical document, which right now is not in the scope, but we will discuss as we go further and further. So once we uh, just a minute, there is there is a question, what is microstructural attribute? So microstructural attribute is nothing but something which can be above the compositions. When you match Q1 and Q2, that is quantitative and qualitative. So apart from qualitative, we go further, and then maybe there are some requirements, something like a critical material attribute. For example, if there is a product where the API particle size D90 is defined, so if you even go and match the particle size also, if there is a not a pattern, then that is called as microstructural sameness. You are trying to match something. For example, one example is that of sucral fed suspension. In case of sucral fed suspension, there is a bio guidance where you have to match Q1 and Q2. Then apart from Q1 and Q2, because the sucral fat acts in your GI tract, the particle size is very important. So in case of sucral fat, when you match the qualitative quantitative composition, you have to match the physical criteria of particle size also. You have to do the sameness study of the API, all the chains and all which are present in the drug molecule, you have to match. So that becomes your Q3 part. That is the microstructural attribute. I hope you got it. So microstructural attribute is nothing but matching certain parameter apart from Q1 and Q2. I think I'm clear now. So now moving uh, to the presentation. So international, I have discussed these guidelines. So the key quality guidelines, Q1A R2 is the stability testing guideline, which is as good as a Bible for a generic development. Analytical procedure validation. This is again important, taken care by the analytical scientists. For formulation development, the guidelines which play a major role is the Q8, which is meant for the pharmaceutical development, the Q9 guideline. So when we say pharmaceutical development, it will cover the QBD part also. When we say Q9, it is the quality risk management, how we manage the risk. Q10, it is the PQS, that is performance qualification system. So right now I'm just enlisting this guideline, and maybe once we walk through the overall development process, maybe I will take you through the complete guideline. One by one, we will discuss maybe 1 month, 2 months down the line when we complete the general R&D part. We will go through this guideline. Then, apart from ICH guideline, as I told you earlier, there is a global implementation adopted by FDA, EMA, PMDA, and more, leading to reduced development costs. So whenever we are developing generic, it leads to reduced development cost, faster regulatory approval, of course, because we get the approval maximum in a 1 year from filing, because it is a abbreviated drug delivery. That's why. Then we get consistent global standards. Uh, then for generics, usually we design the stability study, method validation, and risk assessment. This is something which is expected across all the global guidelines. Now coming to R&D part. So R&D, when we do pre-formulation, Q3A guideline, which is related to impurities, becomes effective. For formulation, as I told you, Q8 and Q9, this is the development and risk mitigation guideline. Then when we go further after the development of the product, we go through process development where Q10, that is the quality system, will become effective. And for Q11, the manufacturing guideline will become effective. For stability, Q1AF protocols is becoming effective, and then analytics, Q2 and Q14. This guideline anyway we will discuss in future. Now let me go through the next slide. There is, okay, I got it. So when we start any development, first thing is that we have to go through a pre-formulation studies overview. We have to go through a pre-formulation cycle. Pre-formulation is nothing but what work we do before going through the actual formulation. So first thing, or the very basic foundation of development, is characterizing API properties to guide formulation strategy and mitigate risk. Might be this sentence might be going a bouncer, so I'll break it into simple words. Whenever we have to develop, before starting the development, we have to understand what is the characteristics which we are looking into the API. What can be the characteristics? Based on these characteristics, we select the API, and so these characteristics basically becomes a guide for overall formulation development. Now, when I say characteristics, we will discuss this when we go through the some advanced sections such as QBD. Right now, just remember that before the development, we have to have a characterization of API based on which we decide what will be our criteria or what will be our requirement for API. So when we see this, what is our objective? So our objective is first to understand the API behavior. So before going through the formulation, we have to understand whether what it is the solubility of your API, what should be the particle size of your API, or what is the degradation pathway of your API. So basically the behavior or the uh, critical material attributes of the API needs to be understood. Once we identify that, we have to identify our formulation risk. Means whatever formulation we are going to develop, based on the API parameters and our QTPP, what can go wrong, that we have to identify. And then we have to go through the excipient selection. So this is, if I say in a very crude language, this is something called as your literature search. And mind you, this is just a very basic thing. actual literature search and all, we will be having a practical sessions once we complete this presentation. As of now, just remember that literature search is nothing but getting the information mostly about the drug properties, drug behavior, identifying the formulation risk and the excipients. So usually it takes 3 to 6 months for this phase to go. That is the literature search phase. So in the literature search phase, these are the details which we see. In physicochemical details, we study the whether the API is soluble, insoluble, or having pH dependent solubility. That is, whether the API dissolves at certain pH and remains stable at some other pH, or does not dissolve at certain pH, or the pKa value we identify. We try to identify the polymorph used by innovator, because the polymorph again determine your bioavailability as well as the performance of the drug. So this is important. Then you have to understand the chemical stability of the drug. How stable is your drug? How does your drug behave in stability? Before the API comes, the API supplier or manufacturer has already done the stability study, 6 months accelerated and maybe long-term study, and they have submitted their DMF to the FDA and FDA approves. That data you can get from the innovator from the open part of DMF. From that open part of the DMF, you have to gather the data, and this data you have to study properly that how this API stability will impact your formulation. Then of course particle size is important for bioequivalence. We will discuss this when we do the actual literature search. Then apart from these characteristics of API, you have to also understand the analytical method. What is the method, whether it is HPLC or some other method? It can be a UV method, it can be a mass method, but mostly it is HPLC method. You have to identify the XRPD to identify the form of API, the polymorph, and whether it is present in clinical, it is present in crystalline or amorphous form. Then DSC to identify the characteristic peak of the API. So these are the physicochemical parameters which you have to go through. Then in case of biopharmaceutical parameters, you have to understand the intrinsic dissolution rate, how your drug dissolves in which pH in water. You have to understand this parameter. And then you have to understand uh, the permeability assessment, the in vivo permeability test and all you have to identify. Then whether there is a food effect or not, you have to identify that too. Then once the BCS classification and all is done, and you have all the information related to the API, you have to go through the risk assessment. So when you say risk assessment, in risk assessment, you have to identify which parameter of API or which critical material attribute of the API can be risky in development of your formulation. For example, if your drug is degraded by oxidation pathway, then in that case, your formulation development has to be in a direction where your product gets least exposure to oxidation, or you have to have some excipient which act as antioxidant. So this risk you have to identify. For example, if your product or if your API degrades in presence of moisture, then in that case, your API or your formulation will be at risk whenever water is present in the formulation. So in that case, either you will go for a dry granulation process or you will go for a non-aqueous process. So this is basically identifying the risk. So the chemical and physical risk, then with any of the excipient, if it is incompatible, that you have to identify, and then the bioavailability challenges you have to go through. Once you identify this risk, you have to mitigate the risk. Mitigation is nothing but decreasing the risk by way of your experimentation or by way of your lab trials. So you have to add protective excipient. As I told you earlier, that if you are adding some antioxidant, it is becoming a protective excipient for the API. And then if you optimize the process, instead of wet granulation, you are using non-aqueous granulation. You are mitigating the risk by removing the water. Sometimes the risk may be because the API is not soluble. So solubility enhancement, again, you can do by reducing the particle size or by adding certain excipient in your formulation, which can increase the solubility of your API. So here are the different API characterization techniques. The spectroscopic analysis. So of course, this will have the UV and HPLC methods. Then you can identify the polymorph by FTIR and Raman, and then the structural conformation by NMR. Of course, when you are developing a generic and when you have got a approved DMF, you don't have to go into all those details. It depends, but mostly for polymorphic form and all, of course, you'll have to do some studies to confirm what polymorph you are going to use. Then the thermal analysis of the API, that is the DSE, where the drug DSE gives a peak at the melting point of the drug, which is the characteristic of drug. To identify it is always helpful. And then the thermogravimetric analysis, that is TGA, where you can get the decomposition profile, how the API decompose or what moisture solvent content is present. This is all about the thermal analysis. Then the most important part for selecting the API is the particle analysis, that is the laser diffraction and microscopy, XRD, and the flow properties of API you have to go through. Then the stability and purity. So for stability, so for example, whether to determine what is your degradation pathway, there are two ways. You have to follow both the ways. First way is if you get the open part of DMF from the vendor, you get the forced degradation studies where your API is exposed to higher temperature, that is the thermal study, higher humidity, or to light, or to oxidative stress, or to different pH conditions to determine how your drug behaves there. And based on this data, you can determine or you can decide how your formulation is going to be developed. This is nothing but forced degradation studies. And of course, you do the impurity profile to identify which impurities are present, because there are guidelines that your impurities should not go on a particular level. This we will be discussing when we go at that stage. So now coming to the physicochemical property analysis. So now since now you have the API, so what are the critical properties of API which you will study? First thing is the pH solubility, whether your drug is having pH solubility, at what pH it solubilizes. This is important because depending on the pH solubility, you will have to identify your dissolution media, you have to identify or you have to make your drug or formulation in such a way that at that pH the drug gets released. And sometimes, depending on the pH solubility, it is having impurity generation, which you have to study. Then the co-solvents present in the API and the temperature effects you have to study. Intrinsic dissolution rate is nothing but the how your drug itself dissolves without the presence of excipient. That study you have to see. Uh, then the physical properties such as the polymorphism, crystalline form you have to study. You have to see hygroscopicity, how hygroscopic your drug is, what is the particle size, what is the thermal behavior, all these critical parameters you have to see before starting the formulation process. And simultaneously, as you start with the development of the formulation method, the analytical scientist will start working in analytical method. So the analytical scientist will check the solubility, they will check the stability, polymorph, particle properties, and all these properties will be checked simultaneously by the analytical scientist also. So now coming to the formulation implications. So in formulation implications, uh, if your API is having low solubility, how you will develop the formulation? So of course, you will try to have a API with the highest reduced particle size, that is the micronized API. You will achieve by some of the technique, either by micronization or by solid-solid entrapment. These techniques we will take or discuss when we go through the practical sessions. Right now, just remember that if your API is having low solubility, you will reduce the particle size, or you will go through the spread dispersion where your API will be dispersed on certain dispersants, or you will or you will add lipid in the formulation which will increase the bioavailability of your formulation, or you will formulate into a self-emulsifying system. So this is related with the low solubility. Now suppose if your API is having chemical instability, then in that case, how will you determine or how will you stabilize the API, or what you will do? So if your API gets degraded in a particular pH, you will modify the pH, as simple as that. So for example, the classic example is of omeprazole. Omeprazole gets degraded in your stomach acid or in your acidic pH. So that's why we see formulation of omeprazole as enteric release. That is, the drug doesn't release in the stomach and it gets only released in presence of basic drug. So either this way, or if you add sodium bicarbonate 500 mg, it will neutralize the acid pH in the stomach. The pH will come to neutral and your omeprazole will not be degraded. So this is what is meant by pH modification. Then of course you can add antioxidant. So when you add antioxidant, so if your drug gets degradation by oxidation, you add antioxidants like butylated hydroxyanisole or BHT, butylated hydroxytonyl, and it will break the oxidative pathway and it will stop your drug from degrading. Similarly, when the moisture is very high in your formulation, so what you will do? You will either target a low LOD value or non-aqueous or dry granulation process. That is where we address the chemical instability. And if your drug is light sensitive, you will add you will perform the all processing in sodium vapor lamp. You will store the drug in black polybags and all. So in this way, you will address the photolytic degradation of the drug. So entry coating, I already told you, if the drug is getting degraded in HCl, formulated into entry coat so that the drug is not released in the zero point 0.1 normal HCl, but the basic pH of your intestine. Then if your drug gets degraded by the headspace, by oxygen present in the headspace, mostly in case of bottle dosage forms of ANDA, replace the oxygen by nitrogen. That is also one way of addressing. Then we have discussed low solubility, we have discussed the chemical instability, then we come to the physical instability. So physical instability is nothing but the polymorph stabilization, stabilizing the polymorph. By what technique you can stabilize the polymorph of API? Amorphous stabilization, crystallization control. These are all the physical characteristics of API which you need to take care. This we will discuss when we go to that place. Then let me move to the next slide. That is excipient selection. So guys, this is the eighth slide and almost it is 9 minutes to 10. So I will take you through this slide for 5 minutes and then we will stop here and then remaining slides we will look into other area and remaining last 5 minutes we will discuss if there is any question. So how do we select excipient? So we will take this briefly and then when we go through the literature search, I will take you again through the detail. So when we select the excipient, it is by the QBD approach. QBD is nothing but quality based design, wherein the excipients are selected based on the risk. And then the criteria for selection of excipients are the functional performance of excipients, that is disintegration and dissolution, how you want the disintegrant, how you want the dissolution. Based on that, you will select the disintegrant. Binding and compression. So binding is nothing but binding the granule. So how we will select the binder. And if your formulation is having flow problem, how you will improve the flow by use of glidant and lubricant. This is the way how we select the excipient. Then the quality attributes. So quality attributes basically, if roughly it will be the material attributes, that is API compatibility and stability. So API is compatible to which excipient? That is the material attribute you have to see. Then the processing robustness you have to see. And then the regulatory compliance you have to see. So this part, anyway, we are going to discuss once we go through the QBD section. And then cost optimization. So any formulation, however good you develop, but if it is of very high cost, it is useless. So cost optimization should always be in your mind. So now coming to the excipient classification. So there are different type of excipient in a formulation dosage form. This is for solid, liquid and injectable. Once we complete solid, we will go through liquid part also, but right now not in the scope of this tutorial. So in solid, the diluents which are gives the mass to the solid dosage form or the tablet which is present in very higher quantity, and the classic example of diluents are the lactose, MCC, mannitol. Then the disintegrant which brings about the disintegration and then subsequent dissolution. These are the crasscormelose and sodium starch glycolate. Binders which binds the granules, which gives strength to your granules for compression. These are polyvinyl pyridine, hydroxypropyl methylcellulose, and starch. And your lubricant, typical example for lubricant, lubricant is nothing which gives a lubrication on the compression machine. So that sticking is not there, ejection is smooth, and tablet gets ejection properly. Most commonly, 99% use is max stearate. Then you have other lubricants such as calcium stearate, stearic acid, are also lubricants. So these are the different type of excipients. Then the excipient risk assessment, high risk, medium risk, low risk. This we will be discussing anyway when we go through the QBD section. So I think it's almost now 5 minutes to 9. So I will stop this presentation here and then in the next session we will start with slide number 9. I will stop this sharing and maybe if you have any questions, you can start discussing with me. I will answer for next 5 minutes and then hopefully we should be able to meet on Wednesday or Thursday. I'll inform you in advance. So any questions? Any questions? Anything related to the pace or you want anything specific from me, you can freely write over here and you can get in touch to me on LinkedIn. Of course, I have already received lot of questions on LinkedIn. I'll take time, but I'll respond to everyone. Everyone is welcome. So any question? No questions? So I'll wait here for 2 minutes. If there is any question, or then I will close the session and maybe we'll meet next time. And this video is already recorded. So let me upload it on some suitable platform and then I'll upload the link on LinkedIn. So you people can go through the LinkedIn and refer to it if you require anything. So I think since there is no question and almost time is up for today, or wait, am I hanging? Was the presentation completed? I don't know because I see myself as hanging. So was the full presentation there? Okay, there are two messages. Let me see what is it. Sorry, I'm not able to see the message. Thank you for the good session. You are welcome. I'm able to read only one comment now. Let me see if there is any way I can read all. Could you please share your slide in LinkedIn? I will share these slides maybe uh, once the session is over together, maybe after 8 to 10 days, I will share it. This was Rajiv. So Rajiv, I will definitely share it, but problem is that LinkedIn is not allowing higher memory presentations. So maybe once this is done, I will break it into separate slide and I'll share it. Then there is a message from who? Francis. Where did the message go? Where did the message go? So Mr. Francis is asking for complex injectable. So let us first complete the tablet section, solid oral. Then we'll start with injectable. So it will of course come, I think, 1 month. We'll take 1 month for solid oral. Then we'll start with the injectable. I have very good presentations for injectable also. So once we reach there, we will have a full dedicated session on injection as well as liquid and topical. All this I am going to cover. Don't worry. I think there is no further question. So I think it is good time to end this session. Maybe I think there is one more question. If please arrange session on B study, of course it will come now, but it will come only when we go on that phase. This is basically session is meant for freshers, just out of college or M.Pharm and B.Pharm students. So initially it will be a basic session. Once the engagement level increases, we will go for B and all. Don't worry, I'll take that session also. But it will take some time to go through that stage. I'm organizing this session so that the freshers who are coming and then they face problems with the jobs and all. So if they at least they have this knowledge, it will help them in gaining the or passing the interviews, and also I'm arranging it for this. BE sessions also definitely I will take. I have a good expertise on BE sessions. So maybe once we reach towards the end of the development, we'll start with the BE sessions. I hope I have answered this, and then waiting for the juniors. This presentation was good. Only the live streaming was quite lagging up behind, although it was quite basic. It will still. Also, I told you in the beginning itself, it is something very basic for the students and not for experienced people. Real experience discussions will come over the period of time, not right now. So I think with this, I will end the session. Thank you. Thank you so much for your time. And let's see what we will do. Uh, so Asim Kumar, it is basic. It is quite basic because it is meant for freshers and not for experienced people. More detailed presentation will come when we reach some heavy stage, not for the beginning as of now. I In the beginning itself, I informed that this is meant only as a guide for the freshers. Advance I will take once I reach there. And regarding the live streaming, let me work it out. I'm also totally new. So let me work it out how to improve it, but I will definitely try to improve. Okay, thank you. So with this, I take your leave. Good night. Take care. Have a nice weekend tomorrow. Bye bye.